All right. Welcome, everybody. You are tuned in to Science Café. This is an event that's put on by the Conservation and Research Department at the Atlanta Botanical Garden. We're really excited to have with us this evening Namrata Pradhan, who will be presenting on seed viability testing for the research and conservation of epiphytic and terrestrial orchids. A little bit more about Namrata. Namrata was born and raised in India. She obtained her master's in zoology from Loyola College in Chennai, India. She worked on the synergistic efficacy of 3 plant extracts, Virginia ciliata, Acorus calamus, and Dioscorea bulbifera, for antimicrobial activity for her master's dissertation. She is currently pursuing her PhD from Guangxi University in China. She received the Zhenwu Scholarship from Guangxi University to do her PhD, which focuses on improving orchid seed conservation efficacy using various in situ conservation tools, such as cryopreservation, seed viability testing, germination, and morphology. And she also serves as the editorial assistant of the Journal of Sustainable Forestry. Before we get started, I just want to let folks know that we are recording this event. So if you have friends or family that were unable to attend, we will have that posted on our website later, and I'll have that link for that at the end of the talk. Also, if we are going to have a question and answer section after Namrata is done speaking. So if you have any questions, just look at the bottom of your screen there and you should see a little Q&A feature. So you can go ahead and type questions in there and we'll do our best to answer as many of those as we can. And actually, Jason Ligon, who is our seed bank curator, is going to be moderating that Q&A portion. So. But yeah, without further ado, Namrata, thank you so much for being here and welcome, and I give you the floor.
Thank you so much. Thank you for the introduction. Let me share my screen. Is that good?
Yeah, looks great.
Okay. Okay, thank you again for that introduction, and thank you for inviting me and giving me this lovely opportunity to share my research with you all. So I am Namrata Pradhan. I'm a PhD student at the College of Forestry in Guangxi University, Southern China. And today I will be talking about a part of my PhD research titled Seed Viability Testing for Research and Conservation of Epiphytic and Terrestrial Orchids. So this is a brief overview of what my talk is going to consist today of. As you can see at the top, there will be, so there is going to, okay. So there's going to be a brief introduction about myself and my research. Then I'm going to highlight the knowledge gaps in this field, followed by the objectives of my research, the methods, the results, the conclusions that I obtain, and the novelty, and finally end with acknowledgments. So before I delve into the seed viability research that I have been doing, I just want to briefly mention as to how I came, to be where I am now. So I belong to this small but beautiful hill station in Northeast India called Darjeeling. And you may be familiar with Darjeeling with the Darjeeling tea, because that's what Darjeeling is globally known for. And I did my entire schooling from Loreto Convent School, Darjeeling, as you can see in that small picture. So it's a cold region, and it mostly snows during the winter months. And this winter it was especially a heavy one. So From that cold northeast region, I then had to move to the forever hot South India, where the climate is relatively hot most of the time. And I went there to pursue my master's in science in Loyola College, Chennai, under the supervision of Dr. Alvin T. Fleming, who was then the head of the Department of Advanced Zoology and Biotechnology. There, my MSc dissertation was focused on the synergistic efficacy of 3 plant extracts, Bergenia ciliata, Acorus calamus, and Dioscorea bulbifera for antimicrobial activity, which was later published in 2016. So I did my master's there. I finished there in 2016. And then in 2017, I went from that hot tropical southern India to a somewhat cooler subtropical southern China to pursue my PhD. So I joined for my PhD in the College of Forestry in Guangzhou University in the Regeneration Ecology and Seed Biophysiology and Conservation Laboratory Group that was headed by my supervisor, Dr. Uromi Managi-Goodale. And since 2017, I have been working there as a PhD student, focusing on improving orchid seed conservation. And I'm currently in my final year. So I have to mention one of the few cornerstones of my PhD journey was when I got this opportunity to go to Kew and present there. Just a second. Let me just move this. Okay. So then I went to Kew and presented a part of my PhD research there too, which was titled Seed Viability Assessment of Cymbidium species and their conservation using cryopreservation, which was with the support of the Lennox Boyd Trust Bursary Award that was given to me by Kew. And I went there with my PhD supervisor, Dr. Uromi, and it was a presentation at the 7th International Orchid Conservation Congress in Royal Botanic Gardens, Kew. And it was a milestone because I could experience, I went to the seed bank there and I got to experience how the researchers have been working so enthusiastically and passionately towards saving this fascinating species. So with all this experience from the trips and through my own trials and errors, I then started working towards improving orchid seed conservation. So let me now delve into the species that I'm passionate about, Orchidaceae. We might already be knowing that Orchidaceae is one of the largest plant family. In fact, the second largest plant family after Asteraceae. It has more than 25,000 species and 200,000 currently identified hybrids. Of course, the number keeps increasing every year as new hybrids, like hundreds of new hybrids identified each year. And orchids are distributed everywhere except Antarctica. And orchids also diverse in their habitats. We have epiphytes and then terrestrials, there's lithophytes and some aquatic as well. And the most challenging part of working with orchids is the orchid seeds, because they are the microseeds or the dust seeds. They are very, very minute. They are numerous, but they lack the storage reserves also. So because of their size, they are actually very challenging to work with. Orchids are known worldwide for their numerous uses in medicine, ethnobotanical uses, ornamental, and ecological. Ecological because, if you have an orchid in your backyard, or if you have an orchid in the place where you work in, you will understand that you are actually working in a balanced ecosystem. Because if there is any disturbance in the ecosystem, then the orchids will be the first to die out. So in case next time you see an orchid dying, in outside in the nature, I mean, of course, not in the pot, then you might understand that they are serving as the bioindicators that there's some disturbance in the balance of the ecosystem around you. So they're excellent bioindicators. And of course, we know that there is extensive uses of orchids in medicine and even for perfumes and ornamental. Of course, we all would like to have an orchid pot in our rooms, right? So because of all of these uses, especially the commercial uses, orchids are illegally collected. They are over-harvested to the extent that the entire family is now highly threatened. So we need efficient conservation measures for orchids. It is now known that orchids need protection because the entire Orchidaceae family is listed in the Appendix 1 and 2 of CITES, which is the Convention on International Trade in Endangered Species of Wild Flora and Fauna. Orchids is also the family that has the most species as threatened in the red list of IUCN, the International Union for Conservation of Nature and Natural Resources. And of course, there are several species in the different national red list, such as the Chinese red list. So there is a clear need for efficient conservation measures if we are to reap benefits from this fascinating plant family. So what kind of conservation is good? Well, in situ conservation, which is conservation in the natural habitat, is of course the best option for safeguarding these diminishing species. And just in case you're wondering, I'm actually examining an orchid seed pod in the Yachang Orchid National Nature Reserve here. It is fortunately my study site as well, and one of the very few areas in the world that is actually dedicated to orchid protection and also regarded as the orchid hotspot. So yeah, in situ conservation is the best option. However, due to increased habitat loss, fragmentation, and habitat degradation, we need to opt for ex situ conservation measures, that is conservation outside the natural habitat. And one of the best possibility for ex situ conservation is seed banking for future use. Here I was trying to seed bank some of the orchid seeds that I had collected from the reserve, and trying to cryopreserve them using liquid nitrogen. Yeah, so why conserve seeds? I mean, why seeds and not other tissues of the plants? What is the advantage? We know that seeds preserve the plant genetic material. Seeds are abundant, and this is especially true if you're working with orchids, because as you can even see in the picture, it's a very tiny seed pod that I have collected, but when you open it, there's like thousands of seeds inside. So because they are so minute, they are also numerous. That is the trade-off that they have. And seeds have the ability to regenerate fully grown plants. We well know that. And seeds have a lower cost for storage. And this is again especially relevant when you're working with orchid seeds, because they are so minute that thousands of seeds can be stored within a small storage facility. So it's actually economical when you're seed banking the orchid seeds. So let me just define for you guys what I'm basically talking about, the two main terms of this talk: seed conservation and seed viability test. Seed conservation or seed banking is ex situ germplasm storage, or in simple words, seed storage for research, conservation, and future reintroduction to the wild. And an integral and very, very essential part of seed conservation is seed viability test. It is a test to assess if a seed is alive, metabolically active, and possess enzymes needed for germination. So what is the use of seed viability testing and seed banking then? Seed banking is an important conservation tool And seed viability test in seed banking is important because it is needed to assess whether the seeds are alive and usable after collection or while being in storage. So it's simple, right? I mean, when you're banking the seeds, you don't want your seeds to be already dead and not know about it. You want the seeds to be alive while they are in storage, and that can be facilitated if you have a reliable seed viability test that can be routinely used. So that is why a seed viability test is an integral part of plant research and conservation. So the success of seed banking is actually determined through these routine seed viability tests. And no wonder we thus require the selection of an appropriate and reliable seed viability test if we are to have a successful seed conservation. So what kind of seed viability test would we then use for seeds as small as orchids and as challenging as them? So as you can see in the picture that I have put, I was actually extracting the seeds of the orchid Cymbidium thracianum from the seed pod in the laminar flow cabinet. And you can see how tiny they are. Like they are literally what they are categorized as dust seeds. They're literally dust seeds. You can see there's thousands of seeds there coming out, and they look like dust. So therefore, the viability tests that are normally used for other species like the cut test, the X-ray test, or the excised embryo test, those kind of tests, it's not practical to use on orchid seeds, because they are so minute that it's not possible for you to just cut and, visualize the internal tissues with your naked eyes. It's not practical when it comes to orchid seeds. And therefore, we need to opt for chemical-based test when it comes to orchid seeds. And the 3 chemical tests that have been so far used on orchids is the tetrazolium test, or the TTC, fluorescein diacetate test, or the FDA test, and the Evans blue test that I will refer as EB test henceforth. So let me just briefly tell you about the principle of these 3 tests. So the TTC and the FDA test are supposed to be the enzymatic test, which means that they rely on specific enzymes in order to give the results of the viability when you're testing. So the TTC test relies on the dehydrogenase enzyme, which means that if your seeds are alive, they are supposed to contain the dehydrogenase enzyme, and the enzyme is supposed to be functional, which will then allow the staining to occur. So the colorless solution turns to the red or orange color solution in the seed, and that's how you know that the seeds are viable. When it comes to FDA test, it relies on the esterase enzyme, which means that if your seeds are active again or alive, they will have the esterase enzyme functioning. And it's because of the esterase enzyme, it converts the FDA solution to fluorescein, which gives out the fluorescence. So suppose your seeds are emitting the fluorescence, it means your seeds are alive, Now, unlike these 2 tests, the EB test, or the Evans blue test, is a non-enzymatic test, which means that the EB test does not rely on any enzymes to function. Rather, it relies on the membrane integrity. Let me explain that a bit. So it means that suppose if your seeds are not viable, which means if they're already dead, they're going to have a corrupted cell membrane. So it means that your cell membrane will be damaged, and the Evans blue solution is actually a non-permeating dye. So suppose it permeates inside the seeds, it means that the cell membrane was not intact, it was damaged, which would then mean that the seeds are not viable. So suppose if you have staining in the seeds after you do this test, it means that your seeds were dead or it's not viable. So actually, it has been said that the non-enzymatic test is actually more reliable than the enzymatic test. And the reason being that sometimes, the enzymes can still be persistent even after the cell death. So suppose if you're using such seeds that are already dead, but just in case the enzymes are still persistent and you're doing your viability test at that time, you can actually have errors in your result because you will basically get a false positive result. So in contrary, the EB test is the test that actually gives a direct indication of cell death because it is not dependent on any enzymes. So with that, let me tell you about my review. So after I learned about these tests, I wanted to see what's happening in the world of orchid seed viability testing and why some of the few papers that I read are actually saying that TTC test is not reliable. So I wanted to see, is that the scene overall so far? So I reviewed about 100 studies that included either of the three orchid seed viability tests, tetrazolium test, fluorescein diastase test, and Evans blue test. And it was clear that I found that the most commonly used seed viability test in orchids is the tetrazolium test, because out of the 100 studies, at least 93 studies had mentioned tetrazolium test. I mean, had included tetrazolium test in their studies. However, unfortunately, majority of the studies reported the tetrazolium test to be unreliable and inconsistent. Actually, there was 83% of the studies that actually had indicated whether it's reliable or not. So out of the 83%, about 64.5% of the studies said that it is not reliable, it is inconsistent, and we cannot rely on the TTC test. So I would assume that even though there has been all these tests saying, claiming the TTC test to be non-reliable, we are still using it because, it's fairly simple, one, and two, it's still mentioned in the international seed testing rules, and those rules have not changed for more than a decade. So since there's no change in the rules and the rules still ask for the TTC test, the researchers continue to use that, even though it's said to be inconsistent. So there was clearly this knowledge gap that the most commonly used seed viability test in orchids is the tetrazolium test or the TTC test, but it is reported as lacking consistency and accuracy in most studies. Therefore, there was a need to determine a reliable seed viability test for orchids of different life forms like epiphytes or terrestrials for their efficient research and conservation. Now, in order to achieve that, I aim to determine a reliable seed viability test for orchids of at least two different life forms, epiphytes and terrestrials, and to determine the factors that are most influential in orchid seed viability testing. So let's move on to the methodology. In order to achieve those aims, I investigated 20 orchid species, which included 5 epiphytic and 15 terrestrial. All of these 20 species, except for one, is red listed in the Chinese red list. And this list also includes 2 species that are red listed in the IUCN as endangered and vulnerable, Paphiopedilum dianthum and Paphiopedilum hirsutissimum. So I collected the matured seed pods of these 20 species from Yachang Orchid National Nature Reserve, located in Guangxi province in southwestern China. So southwestern China, which includes the Guangxi province, is actually one among the 9 world orchid hotspots. And the Yachang Orchid National Nature Reserve is actually one of the few areas in the world that I've already mentioned before that is solely dedicated to in situ protection of orchids. This area is also high in orchid diversity because it has about 147 recorded species of orchids belonging to 44 genera. So it is often regarded as an orchid diversity hotspot as well. So I was fortunate enough to actually have that region as my study site. And Yeah, that's where I collected the seeds from. Now, let me just mention here, when you are seed banking or testing the seed viability, it's always advisable to collect mature seed pods. And the reason being, these matured seed pods is said to have the highest viability when it is at the state of the highest physiological maturity. And the reason is that it's because when they are at their highest physiological maturity, they have the lowest moisture content, and therefore they can be desiccation tolerant. And of course, they have high abscisic acid, they have high the lipids and protein deposits. Because of all of that, they have the maximum storage potential. So therefore, when you collect the matured seed pods, you know that they will have a long seed longevity while being in storage. So make sure that you're collecting the matured seeds. And the one indication of your seed pods being matured is if they are at the verge of dehiscence. So when you go out in the reserve, and if the seed pod is at the verge of dehiscence, which means that they're just opening, that's when you know they are matured, and then you collect it. So then I collected the seed pods, and then I brought them to the seed biophysiology laboratory in the Forestry College of Guangzhou University. And then I extracted the seeds under the laminar flow cabinet to maintain the sterilized environment. And after extracting the seeds, I then dehydrated the seeds in vacuum dryers at less than 10% humidity using anhydrous calcium chloride and maintained about less than 10% humidity for until 5 days. So after dehydrating the seeds, I put the seeds in sterile sealed cryo vials, and then seed banked them at minus 18 degrees Celsius in the freezer. And then I use them for these experiments within 30 days of collection to get the fresh set of the seeds. And I also took the seeds that were banked for a long time, for 6 to 8 years, to get the long-term storage subset. And then I subjected these seeds to 3 seed viability tests. The 3 most commonly used seed viability tests: tetrazolium test, fluorescein diacetate test, and Evans blue test. So it's TTC, FDA, and EB. Now, all of these 3 tests undergo 4 common phases: sterilization of the seeds before the experiment, pre-moistening with distilled water, incubation of the seeds with the different chemicals that is used in the test, and then evaluation of the viability under the microscope. So I also did another round of these tests without sterilization of the seeds, because I wanted to see the effect of sterilization. And so each species had these 3 tests undergoing with and without sterilization. And for each treatment of each test, for each species, I used 4 replicates. So there was about 480 viability tests that were conducted in this research. So let me move on to the methodology for each test just briefly. So yeah, before I go on to the individual methodology, let me just mention here that one hack that was useful to me while doing these viability tests with orchid seeds was if you use a disposable syringe while doing these tests. Because, you know, the seeds as small as orchids, they can be challenging to work and even more difficult if you're using big utensils in the lab, like the conical flask or the filter paper and all of that. So it's actually useful if you take the disposable syringe and then fit a 45 micrometer mesh at the top so that it's easy for you to sterilize the seeds or wash the seeds. And I used 1 syringe for 1 replication. So there was like hundreds of syringes. But that's fairly easier than using these big utensils. So with that, let's move on. So the tetrazolium test, so I sterilized the seeds with 4% calcium hypochlorite and 1% V80 for 20 minutes. And before I move on to the next step, it's important to mention here that the seeds need to be really washed very, very well after sterilization, because you wouldn't want the sterilizing agents to be destroying the embryo or killing the seeds. So it's really important. So I wash the seeds with distilled water like about 5-6 times, or sometimes 7 to 8 times, to remove the excess sterilizing agents. And after that, the seeds were soaked in distilled water for 24 hours. And the next day, the seeds were incubated in 1% 2,3,5 triphenyl tetrazolium fluoride solution for 24 hours at 30 degrees Celsius in the dark. So this reaction is is facilitated in the dark. So make sure the seeds are in the dark. The other thing you can also do is cover the syringes with the aluminum foil so that the seeds are actually literally in the dark as well, and not just the chamber. And then after the incubation period is done, I'll wash the seeds once with the distilled water, and then put the seeds in the microscopic glass slide and examine it under the stereo microscope. So under the stereo microscope, I just captured images of it, because then I took those images to image J, and that's where I scored them. The embryos that were stained red or orange was considered as viable, and the embryos that are not stained as non-viable seeds. That's because if your embryos are staining red or orange, it means that they have the dehydrogenase enzyme. That means they are still alive. They still are undergoing respiration. So the stained embryo seeds are the viable seeds. So let's move on to the next test, which is the fluorescein diacetate test. And since I have already explained it in more detail in the TTC test, I will just go through this. So again, sterilize the seeds with 4% calcium hypochlorite and 1% V80 for 15 minutes. And then thorough washing of the seeds again for many, many times. And then soak the seeds in distilled water for 16 hours. And after that, prepare a 0.5% solution of fluorescein diacetate in acetone, and then suspend the seeds in that solution for about 15 minutes. After which, wash the seeds once, and then put them in the microscopic glass slides or a petri dish, and then observe it under the UV fluorescence microscope. And this time you need a UV fluorescence microscope. Stereoscope is not enough, because you need to see the fluorescence. And then the images were taken to imaging, and the embryos that are exhibiting fluorescence are actually considered as viable, because that means that the esterase enzyme is working and giving out the fluorescence. And finally, the last and the third test is the Evans blue test. Here again, they undergo sterilization sterilization for 10 minutes, and then and then thorough washing again, then soaking the seeds in distilled water for 16 hours, and then incubating in 1% Evans blue solution for about 1 hour. Not about, exactly 1 hour. And then after that, washing the seeds to just remove any excess stain, and then evaluation under the stereo microscope. The unstained embryos are viable. So unlike the other two prior tests where the stained embryos were viable, here the unstained embryos are viable, because as you may remember that I told before, if the embryos are stained, that means that even the non-permeating dye, the Evans blue solution, has penetrated, which means that the cell membrane is damaged, and therefore your cells are dead. So this would give a much clearer picture of the evaluation phase. So here I have used the Cymbidium floribundum seeds, and this is the evaluation phase after each of the tests. So the first is the Evans blue test. So as you can see, the seeds are clearly stained blue, so they are non-viable. For viable seeds, they are unstained. For FDA test, the absence of fluorescence is non-viable seeds, and if they are exhibiting fluorescence, they are viable. And again, for TTC test, you can see that the unstained seeds are non-viable, and the seeds that are stained to bright red or orange, they are viable. Yeah. So I have already mentioned about the principles, so there's no point going over it again. So this was just to give a presentation how it might look after you do all of these tests. So yeah, another disadvantage of the TTC test, other than being inconsistent, is that usually, you know, when you're working with dark species, it can be so that if you have a dark, so orchids, you know, they are of different types. So some species may have a darker testa or a darker embryo than the other species. So if you are working with the dark colored species seeds, you might find that it is more difficult to interpret, especially the TTC test, because color of the dark embryo can obscure the staining of the TTC test. So yeah, that's another disadvantage, because of which it is difficult to interpret, and that could be another reason why it has been said to be inconsistent. So yeah, so the seed viability test might look fairly simple to, you know, when you just follow the protocols. But let me assure you, it's not all that simple. It has got its own challenges. It's because these seed viability test actually needs a considerable amount of practice, a lot of trials and errors, and a lot of patience, and you have to be familiar with the species under test. That's because, you know, it's not just enough to follow the protocols. You have to actually get the hang of doing these tests. Until you are just doing it on, you know what's happening and how your species are going to react to all of these tests. Until you reach that point, there really needs these meticulous attention to the details, and a regular practice and all of that. So this is a list that I actually extracted from Gosling et al. 2003, which is a chapter on viability testing in this amazing book called Seed Conservation, that is actually published by the Royal Botanic Gardens, Kew. So if you're working with seeds, that might be a very good book to give it a read. So according to the people at Kew, the success of the seed viability test requires good vision, considerable manual dexterity, well-honed surgical skills, great care, meticulous attention to detail, natural aptitude, regular practice, and familiarity with the species under test. So yeah, so we used the beta binomial model, which was the best fit to our data. And this is the full model that has the seed viability test, sterilization status, life form, that is whether it's epiphyte or terrestrial, storage status, whether it's fresh or long-term stored, and the interaction between viability test and sterilization status as the fixed factors, the species as the random factor, and the response is the probability of finding viable seeds, or the seed viability. And the analysis was carried at 3 levels. First was the pooled analysis, where we kept both epiphytic and terrestrial species together, because we wanted to see what factors affect the seed viability in orchids. And then the life form level, where epiphytic and terrestrial orchids were analyzed separately, and at the species level, where each of the 20 species was analyzed individually. So this is the results. So Here, the PSV is the probability of seed viability. The NS is the not sterilized seeds, and S is the sterilized seeds. And EB is the Evans Blue test, FDA is the fluorescein diastase test, and TTC is the commonly used tetrazolium test. The figure A is for the seed viability test on epiphytic orchids, and B is for seed viability test on terrestrial orchids. So in the pooled analysis, as well as at the life form level, we found that the pattern was similar. We found that sterilization prior to testing, then the seed viability test type and the life form, they were all the influential factors in orchid seed viability testing. And contrary to our hypothesis, we found that the storage status, whether it's a fresh or long-term stored seeds, actually does not influence the orchid seed viability testing. And as you can see here, the sterilization actually lowered the viability especially when you're using the EB and the FDA test. And the results was just the opposite for the TTC test, where sterilization actually increased the viability. And you can see, especially with the FDA test, it causes more stress with sterilization, specifically for terrestrial orchids, because when you do, as you can see, the FDA test has a much lower viability with sterilization as compared to the other test. And this was actually in accordance with the past literature also. Where they said that when you're using the FDA test, it can cause a modest stress on the embryo that lowers the viability. And for both epiphytic and terrestrial orchids, the viability was highest for Evans Blue test without sterilization and lowest for the TTC test without sterilization. And as you can compare the difference, epiphytic orchids generally have a higher viability than the terrestrial orchids. So then we moved on to see how reliable they can be comparatively. So the variance that we found, which is the difference between the maximum and the minimum viability that was obtained, we found that the variance was actually the least when using the Evans Blue test as compared to the variance, the high variance that we found with the FDA or the TTC test, suggesting that the Evans Blue test is more reliable. So in order to see that further, we went on to the species level, and we found that even at the species level, we found a similar pattern going on as the other levels of analysis, the pooled and the life form level. Even at the species level, most species, that is out of the 20 species, 19 species actually showed highest viability with Evans Blue test. And of the 19 species, there were 17 species that showed high viability with Evans Blue test without sterilization, and 2 species with sterilization. And also when it comes to stability, we found that most species showed the least variance when it with the Evans Blue test as compared to the high variance with the other 2 tests. Again, indicating that Evans Blue test is more reliable because it gives less variance with both with and without sterilization. So therefore, we can conclude that the life form of the species and seed sterilization prior to testing is an influential factor for orchid seed viability testing. Evans Blue test is a more reliable seed viability test for orchids as compared to the FDA and the TTC test. And because the storage status, that is whether it's a fresh or long-term stored seeds, the storage status was not an influential factor, was not significant. Therefore, the recommendation of this study to use the Evans Blue test for orchid seed viability testing is actually useful for testing both fresh as well as long-term stored orchid seeds. And this is an important point, especially when it comes to seed banking, because, after collection, you need to see whether they are viable or not. And also you need to routinely see the viability of the long-term stored seeds. So that's useful. So this was the first study to do a comparative investigation of the most commonly used in two orchid life forms, epiphytic and terrestrial. And among the 20 study species, just 5 species had their viability tested in the past, but with just the TTC test. Knowing the viability test that actually gives a reliable viability estimate is important, especially because it helps to minimize errors in the initial steps of the research. That the foundation of your research as well. And also it's useful in ex situ conservation measures, especially the seed banking and all of that, because as soon as you collect, you need to see the viability and to also well-being and storage. That is why we hope that our study actually provides an improvement to orchid seed conservation. So just in case you are interested to know more about this research, it is recently published as a research article in Botanical Studies. So if you're interested, you can download the paper from their site and have a read. So finally, I would like to express my heartfelt gratitude to my advisor, Dr. Uromi Managi-Goodale, for her valuable guidance throughout. I would also like to thank Dr. Eben Goodale for his support in publishing the article in Botanical Studies. And I am also grateful to all our co-authors and collaborators, Dr. Gao and Shuli Fan from Yunnan University, and Dr. Hong Liu from Florida International University. And also I would like to thank all my fellow lab mates. Finally, none of this would be possible without my Juno University scholarship and the funds from Uromi's grants. So before I conclude my talk today, I would just like to quickly mention about this group that I have been a part of for a few years now, the IUCN Seed Conservation Specialist Group, which is headed by Dr. Uromi Managi-Goodale, my advisor, and Dr. Marian Chow, that has been working for years in seed banking from Hawaii. Now, this is a team of very enthusiastic researchers and conservationists working towards seed conservation. So in case you are interested in joining, you're welcome to visit our website, and the link is on the screen. So that's all for today. Thank you so much for listening, and I'd be happy to answer any questions you may have for me, or you can also email me your queries in this email address. Thank you again.
All right, excellent, Namrata. I think I speak for the whole group when I say that I thoroughly enjoyed it. I think that was an excellent presentation and very eminent and useful for people who are working with orchid seeds. So we're starting to get quite a few questions in right now. So I'm going to do my best to just filter through. For all the participants, feel free in the Q&A to keep adding questions as we go through it. And we're going to give it about 10 or 15 minutes just to see how many of those questions we can get through that pertain to the main group.
Okay.
So yeah, so I'll just go ahead and jump in. I think one more general question that someone asked in the beginning, Donna was asking, can the seeds be planted or restored after viability? In other words, is it destructive or not, these things that you expose the seeds to?
Oh, I see. Yeah, so unfortunately, that's one disadvantage, that we cannot use the seeds after the viability test. So it's always suggested that whatever seeds you collect, you're not supposed to do the viability for the entire population, you know. If you collected like suppose 1,000 seeds, you can test in about just 5 to 10% of them, because when you're testing the viability, you're just trying to see if the population is viable. So if you are just randomly taking the seeds from the population and just doing a set of this viability with just a set of the seeds, then you're actually having an estimate of the viability of the entire population. So then you can just use a small subset of the seeds for viability and keep the rest for banking or for germination and all of that. So yeah.
Okay, I think that's excellent. And we also, we talk about storing the seeds long-term. You mentioned banking them at -18 Celsius. So Farelli asks, when you say long-term storage, are you referring exclusively to liquid nitrogen, cryostorage, or other ways? Can you make any statements about different storage methods that may be available for orchids?
Yeah, so there is. Now, especially if exclusively for this study, we just use the seeds that was stored at -18 degrees Celsius. So basically just in the freezer, that was stored with blue silica gel that was used as an indicator of relative humidity. But of course, when it comes to orchids in general, there is a lot of storage options. But again, it's another challenge of using the orchids, because they often show species-specific response to these tests and to storage as well. So there are some species that store well in the fridge, like about 5 to 7 degrees Celsius. Then there are some species that store well at -18 degrees Celsius. And then there is a lot of species that can be cryopreserved. And sometimes cryopreservation actually increases the viability, that I have seen from the past literature. And I have also done another study that using cryopreservation, and found that it's actually true that sometimes cryopreservation increase the viability. So yeah, it is, cryopreservation is mostly more helpful, but again, it depends on the species that you're testing with. You have to first do like a trial and try to see which storage option would work for them best, because they have different responses, you know, to these storage. So yeah, and some may be more desiccation tolerant, some may be less desiccation tolerant. So there needs a considerable amount of trials when it comes to storage also, before you can decide which storage option is best. But yes, it's available, like fridge, freezer, and even the cryopreservation is available for orchids.
That's excellent. I'm so glad that there's so many different options that we have. We do the same thing here at ABG as well. We consider all of those. And I think I'm paraphrasing Judson's question here, but it's combined with a personal question that we had as well. And so they were asking if there are some seeds that are more or less difficult to assess if they're viable. In your presentation and your research, you mentioned how some of the terrestrial orchids sometimes can have darker testes or seed coats. And there was that, it seemed like they were all per test, they were all sterilized at a specific sterilization time. Did you find that with that 20 minutes of sterilization for TTZ and other times for other tests, were there some phylogenies of orchids that were just not as clear or easy to see through the test set?
Yeah, so when it comes to these darker species, yes, it's more difficult. So I had found in one of the past literature, I think in the seed conservation book also from Kew, they have a research on this where they are using the FDA test. So when you're using the darker species that has a dark testa or a dark embryo, then I would suggest going for the FDA test more, because the FDA is not based on the staining, it's based on the fluorescence. So when you are estimating the viability based upon the fluorescent, then it's fairly easier to estimate when it comes to these darker species, because when using the TTC, or even for that matter the Evans blue stain, it's the the staining of the tester or the thing can obscure the staining procedure of the viability test. So yeah, so it's easier to use the FDA test if you're using darker species. In our case, we had the two Paphiopedilum species that were, that had a dark embryo. So in that case, the FDA test was more useful.
Okay. And Raquel also asks, since the sterilization reduces viability, are you considering other sterilization methods other than the one you used?
Yeah, so yes, so there is actually two different views regarding to sterilization. So as you can see with the TTC test, we found that the sterilization is actually increasing the viability. So that has also been mentioned in the literature in the past, that when you're using these hypochlorite sterilizing agents, these hypochlorite sterilizing agents, what it does, it helps in actually allowing the stains to enter inside the seeds. So it actually assists the staining viability test when it comes to these hypochlorite solutions. So suppose if you're using this calcium hypochlorite or sodium hypochlorite for that matter, then they actually assist in these viability tests. That's what is suggested. But then of course, given that sterilization definitely causes stress to the embryo, so there's these two different views. So to have like a common ground, it's always advised that you do like a sterilization trials and see what works best for the species, because again, it depends on the species. That's also one of the limitation of this study, that we didn't do a lot of sterilization trials for each of the 20 species, also because I had a lot of species in hand. But if suppose you're using like fewer seeds or you have more resources available, it's always good to actually do a few sterilization trials and use like different concentrations of agents or different durations, like 5 minutes, 10 minutes, 15 minutes, and see like what works for your species, and then go for it. Because again, mostly sometimes it's species specific, and that has also been mentioned. So yeah, sterilization is important, and trials is also important for it. So definitely, so hypochlorite solutions is said to be better, but then again, you need those trials to actually confirm that fact for your species.
That's great. Yeah, that's great. So there might be a pilot study in the works, and then that makes complete sense versus other techniques with hydroxide or some other way to sterilize. So we have another question about the correlation of your data, possibly. So Melissa McCormick is asking, she's wondering if the tests that you did were ever correlated with percent germination. A lot of times you'll want to see how effective these stains are in comparison to a germination trial. Was that done, or is that being considered?
Yeah, so Yeah, actually, that is a good point, and that's yet another limitation that we have, because I did try the germination, but because of the lack of resources, and also most importantly lack of time, because I was doing this for my PhD, and you know how long orchids take to germinate. Sometimes it's weeks, but then there are some species that take years. So that was not within the time frame that I was allowed, actually. So unfortunately I couldn't now. But yes, definitely, if you have your resources and time in hand, I would definitely suggest that germination be correlated with it. But given that there's also this in this seed conservation book, they have said that often scientists relate viability with germination, but they are like two different things, because viability is just when you see that seeds are viable, and germination is when actually you see an embryonic plant being developed. So you cannot say that a viable seed is similar to a germinable seed. They are different things. Often it's confused, but having said that, I would say that if you haven't had, definitely go for germination, because that gives like a definite estimate of the germinability. Because when you're banking the seeds, you don't just want your seeds to be viable, you also want them to be germinable, for the long run. So yeah, for this study we couldn't, but yes, definitely it should be done.
Okay, that's excellent. Yeah, because I think in seed banking, we ultimately want to be able to preserve that genetic diversity and be able to germinate in the future. So we understand there are always limitations though to what we can do.
Yeah.
Yeah. So Amanda has a great question here. She says that you mentioned that sterilization was detrimental to the seeds for the FDA and EB test. With that knowledge, does your updated protocol for FDA and EB tests not involve sterilization at all?
Yeah, so mostly we saw that sterilization is detrimental when it comes to events flow and FDA test, as she mentioned. So definitely, if sterilization is not, not something that you want in your research, I would say it's better to avoid, because they are causing that stress. But if it is something that, for example, if you want to also germinate the seeds, that you are undergoing the experiments and you want another subset to germinate, so before germinate, I would assume that you want to sterilize the seeds also. So if that is one of your criteria, then yes, go for the sterilization, but you will have to keep it in mind that there will be a slight stress there to the embryos, and not all the seeds might be viable after sterilization. But if it is something that you're considering to do, for example, if you're considering to germinate and go for germination trials where you might want to sterilize, then yes, do the viability test with the sterilization, I would say. Yeah.
Excellent. And this is from Yanni. It was a more general question. They wanted to know if, I guess the staining with TZ is, you were saying that it's difficult sometimes and unreliable with orchid seeds. In your literature review, did you see that TZ is also difficult or unreliable with other non-orchid species as well?
No, sorry. That's something that I did not consider. But yeah, non-orchid species is something I did not review. But that's a good question here. So if you're working with other species, you might want to look up the literature and see what they say. Yeah.
Yeah, I believe that like ISTA protocols and things, they definitely say that it takes a trained eye a lot of times at the family level or genus or even species level in order to analyze seeds of other families. So I think that'd be something to consider.
Yes.
So yeah, I'm scrolling through everything here, just making sure that I've hit on pretty much everything. There are a couple of things I think that you've answered in your presentation. People can go back and see in the recorded presentation, I believe, but I think everything has been answered pretty much. Do you have any closing comments or anything like that that you would like to make?
Yeah, I think I forgot to mention that through this research and maybe the people looking at this presentation, I would also like to just say that through this research, we are urging to the evanciability test to be recognized as the more reliable test than the TTC test, and to maybe help to incorporate it in the international seed testing rules like ISTA, because it is still not incorporated. So and the rules have not changed since many years, and that's why people are just using the TTC test because that's the only test written there. So maybe we can urge for it to be also included and see. And also I would say that not just relying on this research, but also this kind of comparative studies should be extended to a higher number of orchid species, for their efficient conservation, because as compared to this vast plant family, like 20 species is nothing. So, and also through my review, I did not see any comparative studies so far. This is the only one. So I would suggest that to be more comparative studies and see what works for the different species, especially the species that are endangered. Yeah.
Okay.
That's okay.
All right. And there were just a couple questions here I think I overlooked. If we have time for just a couple more. So Caleb is asking about suction. So I believe in order to help uptake the stains, sometimes suction has been used in order to force the seeds to uptake the various stains. Is that a method that you're familiar with or that you've seen in the literature?
I haven't seen it so far, but that's actually a good point. But I haven't seen it in that 100 studies, sorry, that I searched. I did not find suction anywhere. What I did was during my initial trials, I made sure that when I was incubating the seeds in the different chemicals, using the syringes, I made sure that all the seeds were actually suspended in these chemicals, and I was like constantly shaking it, and from time to time. So while they were in suspension, I was like time to time shaking it and just making sure that they are all getting the stain, and none of the seeds are left out. But yeah, I haven't considered suction.
Yes, please get some water. Excuse me, you've been talking for a long time. It's been a great presentation. I think a lot of people are eager to get in a lot of questions. Hopefully I've been able to answer or address everything. And again, if we didn't quite get to it, you'll probably see the answer in the recorded presentation that she's already presented. I think that we're all set. I will hand it back over to Ian just to close it out.
Yeah. Thank you so much for having me.
Yeah, thanks, Namrata.
Yes, thank you so much, Namrata. That was a wonderful presentation, and I know it was very early for you, so we really appreciate you coming out tonight and doing this presentation. And also, thank you to all the participants. We had some excellent questions tonight. Jason, thank you for fielding all those questions. Yeah, it was great. So I just want to let folks know that we did record this tonight, and that recording will be available on our website. And I'll go ahead and send out a follow-up email tomorrow and include the link for that. The easiest way, though, if you're ever curious about upcoming science cafes or want to find old recordings, just Google search Atlanta Botanical Garden Science Cafe or ABG Science Cafe. It'll be the first thing that comes up on your Google search. Also, I'd like to let you all know about next month's presentation, which is going to be with Dr. Christine Folk, who is the Assistant Professor of Cultural Anthropology at Duke University. And she will be presenting on her recent journal article, Ceremony, Medicine, Caffeinated Tea, unearthing the forgotten faces of the North American stimulant Yaupon holly. So that's Ilex fumitoria. Really looking forward to that. Should be a fun talk. And as always, if anybody has any questions, comments, suggestions, feel free to send those to me at isabo@atlantabg.org. Also, definitely check out our website at atlantabg.org/conservation-research/. And we also have an Instagram for any of you folks that are Instagram people. You can follow us at Atlanta BG Conservation. Everybody have a great evening. Thanks for coming out.